Hey guys, welcome to our first lab. So in the lecture, we've been learning a little bit about DNA structure and getting ready to learn more about DNA mutations. Throughout the course, as we proceed, we're going to be learning quite a bit about how to link phenotype to genotype and a lot about how we can use DNA sequence differences to help us understand what's going on with the phenotype. We're also going to be using these genotypic differences are DNA sequence differences to learn how to orient ourselves in the genome. So at the very basic part of all of this, what we're going to need to learn about is markers. So markers may seem a little bit vague at first, but we'll be using this all throughout the semester, particularly in the first few labs, and then learning how to really use these in the last third of the class. So markers, this is really going to be the essence of some of our modern genetics techniques. So we need to know, what are markers? The confusing truth is that this can really be any difference between two individuals. So literally any difference that you can think of. Okay, so what we're going to be learning to do is figure out what differences we actually care about and what markers, what differences can be useful to us. Okay, we'll be working on that throughout the semester. So this can, of course, because it's any difference, include a wide variety of different things and be used for a wide range of purposes. So this includes identification, as you know, like by the FBI. We'll be talking more about that in a bit. This includes paternity tests, looking at species differentiation. So when species differentiated from each other, we can look at gene identification. Something called marker-assisted breeding, which will sort of accelerate, particularly in crop plants, accelerate our ability to get ideal crops and increase yields. This can help us learn more about disease states, developmental stages, and many other things. So these can be used for a wide range of purposes and can be literally any difference. So just to help us kind of orient, let's think of markers as falling into a few categories. These can be morphological markers, biochemical markers, or genetic markers, which is something that we will be really focusing on. Morphological markers would be any observable phenotypic difference. So this would be just being able to tell the difference between two things. Take these flowers, for example. You can probably, right off the bat, name a few differences that you see between the top flower and the bottom flower. So you can maybe note the size of the flowers. You can note whether there's a red ring of pigment around the center or not. But these can be morphological markers. So it's pretty easy to tell that this is a difference. Whether that difference is going to be relevant or not, that's what we're going to be learning to do. Additionally, another example, you can see these little germinating seeds. One of these is a little bit more purple, so looking like it's making a little bit of anthocyanins here, while the other is not. Okay, so you can see, all right, this is a morphological difference. Of course, sometimes these morphological differences are the result of different biochemistry within the cells of these organisms, and even perhaps different DNA sequences. But we can use these just as is under the category of morphological markers. Biochemical markers are kind of what you would think. So this could usually be a protein, like an enzyme, or an RNA difference. So this can be measured in various ways depending on what it is you decide to look at. This could be differences in gene expression. Does one organism have higher gene expression than another? This could be using protein quantification. So you could look at the protein levels instead. This could be using immunoassays or enzymatic assays as well. So for example, you can see some notch expression or wild type versus whatever kind of knockout this is. You don't really have to know a lot about these figures to obviously see that something different's going on with the wild type and this mutant line that we're looking at. So this would be more like a biochemical difference, something that you can observe after doing this type of assay. Genetic markers in some ways are the easiest type of markers, but also the most broad, and sometimes that makes them a little bit vague. Any DNA sequence that's different. Literally any type of sequence difference, period. This could be between two individuals. This could be between different species. This could be anything that's different. So let's just kind of break that down into some potential differences. First, we can have single nucleotide polymorphisms. Okay, so that's a big name for a relatively small difference in a DNA sequence. So this could be, again, a CG pair substituted for a TA pair. So by single nucleotide polymorphism, we literally mean a single nucleotide difference. So you can see that this is going to be a C in Mr. A and a T in Mr. B. Okay, if you're looking at the bottom strand, it's going to be a G and an A. But the main point is this person has one nucleotide base pair difference from this one. And you can imagine if you could sequence this, you'd be able to see that difference and you'd be able to distinguish between Mr. A and Mr. B. So, and these can be exploited in various ways. We're going to be reading more about these as we proceed through this semester and learning how to use these as genetic markers. The main type of genetic marker that we're going to be focusing on in this lab is a type of marker that is called short tandem repeats, or STRs. This is a common name for them. You may also hear them called SSRs, or simple sequence repeats, or I think kind of an older term for this would be microsatellites. So there are a couple of names for this. I'm going to try and stick to STRs for now, but the DNA differences will be the same. So what we're thinking about is, let's just focus in on here. We don't need to know all about the PCR just yet. You'll be able to learn more about PCR in the next video of your assignment. So here, what we focus in, this is the DNA sequence. What STR, or short tandem repeat, means is literally a short sequence, in this case, GATA, you can see it here as well, GATA, that is repeated sort of a number of times, okay? So here we see 4 repeats. Here we have more, okay, than 4. All right, I didn't actually count them, but it's like 12 or 13 or so, okay? So the idea here is that these repeats can be different sizes in different individuals. So for example, I might have 4 repeats giving me a section that is 16 nucleotides long, whereas another individual may have this like 13, 14 repeats that we see here, and that will make us have a much longer sequence. So those type of differences basically boil down to a size difference in the DNA. So we're going to be learning to use these size differences to create different visual patterns on our agarose gels so that we can tell the difference. Okay, so don't pay too much attention to this gel. It's more so that you get the idea that a size difference will help us out. And so if I have a shorter sequence and you have a longer one, by running this assay, you'll be able to tell whether the DNA came from me or from you. And this is a very common one to use, these STRs, mostly because you don't have to do a lot of extra work if you choose these well, because you just amplify them up using the technique that we're going to learn about, and that's it. You can run them on the gel and you're done. Some of these other types of mutations, like single nucleotide polymorphisms, require sometimes a little bit of extra work after the fact. That's something that we have plenty of time to focus on throughout the semester. So again, these products, they're going to be amplified using polymerase chain reaction, or PCR, which we'll learn about in the next video. And then we see the product size. Okay. So specifically in the lab that you're going to be doing is using your own cheek cells that you have harvested on the swabs that you picked up and extracting your own DNA from these. Okay, so we're actually going to be looking at some markers in your own DNA. Specifically, we're going to be looking at a set of markers called CODIS markers. All of these markers are these simple tandem repeats, or STRs, and the CODIS markers are actually those same markers that the FBI uses when they're looking to identify a person. So don't worry, we won't be doing anything where someone could be able to identify you after running this assay. I won't spoil the surprise what we are going to do, but don't worry about that. So what we can see is a list of these CODIS markers, and again, each one of these is one of those simple tandem repeats that we looked at kind of on the last slide, so they're structured the same way. This can look confusing. These names, what do they even mean? Okay, so we've got a lot of these weird names. A lot of times these names are just like an address. They tell you where this lives in the human genome. So I know it's a little blurry, but for example, this one is like D3S1350. I think that's an 8. Okay, notice this is a D3. That means usually it's on chromosome 3 here. Okay, similarly, some of these are a little bit more oddly named, but you can see with these ones that are named kind of D7S, this is going to be on chromosome 7. Okay, D5S, that's going to be on chromosome 5. D8, that's on chromosome 8. So you don't have to memorize that, but check it out. This is just an address. Okay, this is like saying that I live on 1179 Centenary Boulevard. Okay, this is an address in the genome. So don't get intimidated by these names. These are just their addresses. And what we're going to be doing with your DNA is amplifying a subset of these. So specifically, you guys will be using these markers, okay? This will be available, I have this list available to you in your protocol, your printed and posted protocol. So you can see we'll be using PWA, we have the D7, S820, some of these other markers that you can see here, the D3, D13, so a couple of these that we'll be using, we're only going to be using 6, but you can kind of get an idea visually where these would be in the genome. Okay, so again, these are CODIS markers. They can get you caught if you're on the wrong side of the FBI, but that's not how we're going to be using these. So what we're actually going to be doing is using them to make DNA art. So if you've seen those sort of fancy sites where you can do a cheek swab, send your DNA in, and then they come back with something like this. Okay, ours won't be quite this fancy, but you should be able to see some size differences. And you guys will have an opportunity to make whatever type of art you want out of your own DNA. So that's what we'll be doing. The purpose of this lab will be to just get us acquainted with the idea of markers and just give us some practice learning about the polymerase chain reaction, or PCR. After this lab, we'll be doing a couple of more labs. One looking at blood type, we'll be doing some that have to do with GMO organisms, GMO plants, and we'll be using this type of technique, looking at markers via PCR in a couple of different ways so that we have time to really understand what a marker is, how you might use it, and then we'll expand that out a little bit more in the last third of the class.